Okay, so we have talked about how cells go about obtaining energy from glucose, going through glycolysis in the Krebs cycle and electron transport chain. But there are that's how we need to go about getting energy, capturing it and then turning it into ATP. And we do that by actually going out and consuming other living organisms. So you go out and you eat your apple in the morning, it has some good disaccharides in there that you're going to break down or convert into glucose and then run it through your stuff. You have to eat other living things, but not all, as I'm sure you were aware by that point. That apple did not get that by consuming something else, like consuming bugs. Some plants do, not apples. So what they do instead is they're actually able to capture energy from light and convert that into the things that they need. So let's talk about that. Talk about photosynthesis. So just give a brief overview of how this happens. We're going to be using similar concepts to what we were talking about before, especially early on, similar concepts to electron transport chain. And in that, essentially, we are going to energize electrons and use the passage of those electrons to generate or capture, sequester energy in some way. Specifically, the light is going to energize these electrons, and then we are going to move those electrons along some sort of electron transport chain in which we are going to pump protons across a cell membrane, generating a proton motive force. Hey, this should sound really familiar at this point. Basically the same as what we've been talking about already. And like also what we've talked about already, we're going to get some ATP generation from that proton motive force. Very similar to what we're doing as well. We also are going to produce this really interesting molecule called, stop my screen from rotating, actually called NADPH, which is formed by getting NADP, which I'm going to talk about in a moment, and having it accept electrons. Or NAD+, excuse me. There's a plus on there. So, let's talk just really quickly about NADP+. It's very similar to NAD or NADH. Where we have nicotinamide adenine dinucleotide. The only difference here is that the ribose that is attached to the adenine has a phosphate, an extra phosphate on this. So we are, otherwise it's very similar. So continuing on with this brief overview, this is going to run in kind of an opposite direction of what we were talking about earlier with electron transport chain. At the very end of electron transport chain, what happens is that those electrons, eyelashes and eyes, bad things. So those electrons are going to be donated to oxygen, which is then going to react with hydrogen to make H2O. We get water. Take oxygen, turn it into water. We're actually going the opposite direction in this. We are going to rob those electrons from water in this case. So we go from water, and we're going to split it and turn that into oxygen plus protons, and get those excited electrons and run those through electron transport chain. Now that being said, normally you couldn't do this without an input of energy. This is why we dump those electrons into oxygen and make water at the end, because they're fairly low energy at this point, and we're going to just dump this into something where the electrons don't have a lot of useful energy. But we can, because these electrons have been excited by light. We've used light as an input of energy to get those to a high energy state again, and then we're going to use that high energy state to pump our protons, or get protons. Let's talk a little bit more how that works. And then after we we get that, we are going to use this chain to make ATP and NADPH, and what we do is use this ATP and NADPH to take CO2 and turn it into organic molecules. Essentially take it and add it into a pre-existing organic molecule. So this should also sound somewhat familiar. This is essentially Krebs cycle, kind of similar, not exactly, but similar to Krebs cycle working in reverse, where we take organic molecules and we chop off successive carbons, generate CO2, and we get ATP out of it. We're going the opposite direction. We're taking ATP, using it to take those carbons from carbon dioxide and get them into an organic molecule. So conceptually, we are here doing the opposite of Krebs cycle. So diagrammatically, kind of the stoichiometry of this is that we're going to generate ATP and NADPH. So we have those that we've made. We're also going to take some water and some carbon dioxide. Again, we are using water to get these electrons, and we are getting the CO2 from the environment to add into other molecules. And what we're going to get out of that is a sugar molecule. So again here, look at this. We have 6 carbon here. We're going to eventually end up with a 6 carbon molecule here. Again, the opposite of Krebs cycle, where we start with a 6 carbon molecule and generate a net of 6 CO2 per each one of those that we put in. We're going to get 18 ADP plus the 18 phosphates that we've cleaved off of that. And then we're going to generate also 12 NADP plus, and then the 6 protons. So for each glucose that we make is going to require 18 ATP plus some NADPH. Okay. This is all going to happen in this structure called the chloroplast, and we're going to briefly go through the structure of a chloroplast. They're kind of like, in some very broad ways, mitochondria, but we're going to have some very notable exceptions. But one of the things is that we are going to have membranes inside of membranes to create compartments that we can separate protons between compartments and run our ATP synthase with. The difference here, one of the big differences here, is that we don't have these 2 membranes in 2 compartments. We have 3 membranes with 3 compartments. So it looks a little bit like this, where you have membrane 1, membrane 2, and then some other things with membranes on the inside of that. I'll come back and talk about these each in detail, but just kind of to orient you. So first off, we have this thing called the outer membrane. Again, very similar to what we were talking about with the mitochondria, where we had an outer membrane, and just like, similar, not just like, but similar to the mitochondria, this outer membrane is very permeable. It has a porin-like protein. So remember, we had a protein in the mitochondria called porin. We have a protein like porin here that is going to allow a lot of different molecules to get in and out of this, or through this membrane. And we also have enzymes here for lipid synthesis. And kind of intuitively, this outer membrane here is right along the outer cusp here is marked as 1, and this along the outside. And inside of that, again, very similar to what we were talking about with mitochondria, we have this intermembrane space. The area kind of sitting in between the outer membrane and the next membrane in. So then this diagram again is this, so we can get right like this right in here, right in between these two membranes marked in light yellow. And then inside that we have an inner membrane. Now this is one point in which we have a fairly significant difference between the mitochondria and chloroplasts. In the mitochondria, the inner membrane was very impermeable. In this case, it's also very impermeable. I said that's a difference, excuse me. It's also very impermeable here. This is what happens when it's the very early hour of 11 a.m. in the morning. However, we also have a lot of carriers and transporters here as well. So we're still very similar to it. And here is that inner membrane, so sitting right there. And remember in the mitochondria, the area that was sitting inside there was called the matrix on the inside. We have a little bit different name for that in the chloroplast. The area that's sitting inside the inner membrane is called the stroma. And this is going to be the area that is in between the inner membrane and the membrane that's inside of that, which is called the thylakoid membrane, which we're going to talk more about in just a moment. Inside the stroma, we have a lot of different things. We have CO2 fixing enzymes, which we're going to talk more about in a future lecture about how CO2 fixation is happening. We have starch that is in there. We also have, and again, this is another big similarity to the mitochondria, we have DNA, RNA, and ribosomes. So like the mitochondria, the chloroplast has its own DNA that uses to code many of the proteins that are found in the chloroplast. You look at that, that's like all of this kind of darkish yellow stuff that's outside the green but still inside the inner membrane, all throughout here. Okay, now we get to the innermost membrane, which is called the thylakoid membrane, which comes from the Greek word for pouch. It looks like small pouches. It's essentially a flattened sac. Just a weird way to describe it. A group of thylakoids is known as a granum. You know, as a grana, plural, they are all grana. And that they organize themselves into stacks. And those stacks are known as the granum. Talk very briefly about how that, so this right here, this green section right here, this is one thylakoid. They organize themselves into stacks, so this whole stack right here would be a grana. Right there. So these membranes are predominantly protein, about 60% protein. And about 40% lipid. So there is a lot of protein in these inner, the thylakoid membranes. Of the lipids, many of those, about 2/3 or 3/4 of all those, 75%, are glycolipids. Now these membranes have a lot of unsaturated fast fatty acids. which makes the membrane very fluid and means that the proteins in this membrane are really mobile. Can anyone think of any reason in which that would be a good idea? This would be useful. Yeah, how would that help electron transport? Yeah. So we're having an electron transport system here, and if these proteins are really mobile and can interact with each other very readily, it's going to increase the probability that they can pass these electrons on from one to another. So in these membranes we have pigments, such as chlorophyll, but other things sometimes such as carotenoids and other stuff, but predominantly chlorophyll, and some electron transport chain components are here as well. Now the inside of this, we have the thylakoid lumen. This is the interior inside that membrane. Some of those electron transport chain components are actually inside that lumen. They're not going to all be bound to that membrane. That lumen is sitting right inside here in those inside the membrane. Okay. So let's very briefly talk about how we actually go about using the thylakoid membrane and the other components of the chloroplast to capture light. And one of the important components here is photosynthetic pigments, which include chlorophylls, carotenoids, a few other pigments. And these are important because what they do is they're going to absorb light, which will excite an electron somewhere in the pigment. And that electron will go to a higher electron orbital. Now, the consequence of this is that it's not very stable in this higher orbital. It can't stay there for very long. It only stays there for about a billionth of a second. And so something has to happen. And there's really three things that can end up happening. The first of these is that you can get fluorescence. The energy as the electron goes down to a lower orbital can be released as energy, as heat, as light. Generally, when we're talking about fluorescence, we're talking about light as it returns down to this lower orbital. This is all well and good. We use fluorescence. We rely on fluorescence for certain things, especially in science. But if you're a plant and you are trying to photosynthesize, this is not what you want to have happen with the energy that you just captured as light. You don't want to just return it back into the world as light again. And heat is not very useful. Most plants do not run steam engines, so the heat is not going to do it any good. We could transfer that electron to another molecule. So instead of it crashing back down to a lower orbital on the same molecule, it could be transferred to another molecule. Alternatively to this, we could also just transfer the energy to another molecule. That electron could transfer down to a lower orbital, but instead of losing that energy as light or as heat, it could instead be transferred to another molecule instead. So with this background in mind, we're going to start talking about the larger complex in which these pigments are sitting that allow the plant to collect the energy from light. And those are what is known, the larger molecule that this is associated with is called a photosystem, which is a combination of a light harvesting complex that usually has the pigment in it and actually interacts with the light, and a photosystem reaction center, where is essentially how we are going to take that energy that the light has, that the light has transferred to the pigment, and we can then pass it on to other components. So let's talk about these. Let's talk about photosystem 2 first. Now, something kind of funny happened in describing photosynthesis. The first thing in the process is named 2, and the second thing in the process is named photosystem 1. Anyone want to take a random guess why that is?
They discovered them in that order?
Yes, they did, exactly. So they discovered photosystem 1 first. It's called photosystem. And then they discovered another one. Like, oh, hey, we need to number these. This one's 2. So photosystem 2, you have a light harvesting complex, which is called the antenna complex. And this is our rough diagram like we had for electron transport chain, but now we're going into photosynthesis, and this is right here. So we have photosystem 2 that we're going to talk about right now. And this is going to what, this is the, okay, starting over again. This is what's going to interact with light, which is represented here by this little lightning bolt. Lightning and light are not the same thing. I am not teaching you that. But it's represented here as lightning. So we have a little lightning bolt is going to interact with photosystem 2, and then we have some other things. So let's go ahead and talk about what's happening there. Photosystem 2 is going to contain lots of pigment molecules. Again, most notably chlorophyll, and in plants most notably chlorophyll A. These pigment molecules, between 20 and 500 of them, are going to be attached to several polypeptides, generally 3 to 8 individual polypeptides. And this is what's going to actually collect the light. The light is going to interact with these pigments in photosystem 2, and we're going to take the energy from that's collected by photosystem to the harvest complex. We're going to transfer it to the photosystem 2 reaction center. Honestly, I don't really care that you know that's resonance energy transfer, just that the energy is transferred. Yeah?
Why is there such a big variance in pigment?
Couple things. Number one, when we talk about photosynthetic animals and animals that use, or not animals, photosynthetic animals, they actually exist. There are photosynthetic animals out there. They generally swipe chloroplasts from algae, and then they stick the chloroplasts in their tissues and just use chloroplasts and photosynthesize, and get essentially all of their energy from that, which is really cool. For like the things that do this most are nudibranchs. There's some sea slugs. One of them that we know a lot about, there's Elysia chlorotica and Elysia viridis, which they have cool names. The genus of nudibranch Elysia. Go Google it. They look like leaves. They're cool. Okay. Anyhow, okay. So that being said, these systems are in an insanely huge range of life, everywhere from green algae to plants, and plants are incredibly diverse. So we're talking about a lot of different kinds of things. I think there's actually a pretty good variation even within a single organism. I'm not exactly sure why that is, but I do know a lot of this variation is just because phylogenetically it's a huge diversity of organisms to have this. Does that make sense? Yeah. Good question though.
So,
yes.
Photosystem 2, these complexes aren't necessarily like one molecule, it's a family of molecules?
Yeah, well, it's a complex of molecules. It's a family of molecules. So, oh wait, I see what you're saying. So you're asking like, photosystem 2 is not like one individual molecule that's identical everywhere you go look it up, right? No, it's not. It is a family of molecules that fit this function in this. They're all related, but not all identical. Does that make sense? Okay. But then also to the other point is that this isn't like one single molecule. You have various polypeptides, various pigments attached to those polypeptides. You have some special pigments in there, and then you also have a reaction center. So it's all like, there's a lot of stuff. They're all held in kind of close union, but it's not like just one thing. And then Nick, and then, yeah.
Well, back to the photosynthetic animals.
Yeah.
So they're swiping the chloroplasts, basically these photosystem complexes from algae and stuff like that. So I'm assuming that the photosystems that they swiped earlier are expiring somehow. What is not keeping photosystems active on these animals?
Okay, so okay, there's a couple things going on here. Number one, some of the genes that a chloroplast needs to survive, a good number of them, are actually inside the chloroplast itself. So it can, to a certain degree, keep making its own proteins and keep going. The hypothesis here is originally chloroplasts were a cyanobacteria that then symbiotically ended up living inside other eukaryotic cells, and it became more of an organelle than symbiotic organism. So they really do have a lot of what they need to continue functioning and replicating by themselves. However, that being said, they don't have everything. And so if a lot of organisms that pick these up, they will eventually die, those chloroplasts. And so they have to keep eating, a lot of them have to keep eating algae. However, one of these, specifically Elysia chlorotica, it actually turns out that, so I've been talking about genes that come from, like when we're talking about the mitochondria, some that come from the mitochondria, but then some that come from the nuclear genome as well, right? So hypothetically, if you ate a chloroplast, and it's the same thing with chloroplasts, many of them come from the chloroplast genome, but you have some that come from the nuclear genome as well. And so you would be lacking those in most of these cases. Well, actually, it turns out that Elysia chlorotica has some of those genes that it swiped from the alga. It looks like there's been horizontal gene transfer, that nuclear genes that have come from the alga now appear in the genome of Elysia chlorotica, so they can produce some of those factors that are needed to continue the survival of the chloroplast. And actually turns out they can make them survive like a really, really, really long time. So yeah, they have to re-up chloroplasts every once in a while by getting consuming some more, but it's very rare, and they can continue these chloroplasts living inside their own tissues for an incredibly long time. I don't remember exactly. I know it's at least in hundreds of days, maybe 2 years. Keep them operating inside their own tissues, which is really, really cool. Actually, that horizontal gene transfer was just discovered. I was a PhD student, so I'm thinking like 2010, 2009, something like that. That was just published. But interesting stuff, yeah. So they keep them together. So Aaron and them back in here. What's that?
Are typically the same polypeptide used no matter what the pigment molecule is, or does it vary?
You know, I'm not exactly sure. I don't know the answer to that one. It's an interesting question. Good question, Tom. I would guess they'd have to be slightly different because you'd have to, I mean, it's a different molecule that you have to interact with, but I'm not exactly sure. They might be able to interact with almost anything. I don't know. Good question.
On the newborn, at least in chloroplasts, is absent with the chloroplasts?
I believe they are, yeah. They have to, obtain them. It's not vertically transferred. Yeah.
So the photosynthesis provide all the energy the nutrient needs, or do they occasionally munch on something?
I think, yeah, they can provide, especially in Elicea chloroca. If I remember right, this is me going off my vague recollection, so I could be wrong. But I think I remember that it can provide all of what it needs energetically. Yeah. So it just keeps going. Yeah.
What would it do for nitrogen?
What's that?
What would it do for nitrogen?
I'm not exactly sure. Maybe they fix nitrogen. I mean, they are animals, so they're producing nitrogen too. So this is one thing that while the animals are eating stuff, I'm not exactly sure. But I think people would maintain them with only like for long periods of time. Any other questions? Okay. So let's keep talking about other cool things too. Hooray for getting me distracted. I like going off on tangents. Okay. In a typical photosystem, you'd have something that happens kind of like this, where you have a whole collection of chlorophylls. You have a special pigment called a P680, and then you have your reaction center, which is going to then pass this energy, like essentially take transfer of energy and pass this on to other systems. And what ends up happening is you get light interacting with one of these chlorophylls. The fate then is to then pass electrons from chlorophyll to chlorophyll to chlorophyll to chlorophyll to chlorophyll to chlorophyll until eventually it then interacts with the P680, which then passes on to the electron center. So it's not like a really direct route. The energy will continue to bounce around in there until it can reach the reaction center. So let's talk about photosystem 2. Yes?
Every time it bounces around, is it like losing a little bit of the energy, like the heat or something? Is it?
I think it must be, but I don't think it's very significant.
And then what's the purpose of P680? Is that just to like pass it on?
It has to be, yeah, to interact with the reaction center here. It has to, like, P680 is specialized to interact with the reaction center.
And so it's just like an intermediate state because like,
Yeah.
like chlorophyll and the reaction center can interact.
Yeah, that's correct. Okay, so we also have photosystem 2, which consists of about 20 polypeptide chains all from chloroplasts. What's that? Oh, still photosystem 2. Those numberings. Still photosystem 2. What did I say to begin that out? And now photosystem 2? Oh. My bad. I didn't remember what I said. And now more about photosystem 2. What was that? The reaction center consists of about 20 polypeptide chains and all of these are coming from the chloroplast DNA. And it contains a special chlorophyll A molecule known as P680. 680 because this particular pigment absorbs most highly at 680 nanometers. And what this does is you can get energy from the light harvesting complex. So all of those that are in, all those chlorophylls that are within there is going to transfer their energy to P680. The P680 then has an electron raised to a higher orbital. Excuse me, not the P680. The P680 transfers the energy to the reaction center. In the reaction center, you have an electron that's raised to a higher orbital. Excuse me. going to transfer that electron to Plastoquinone. So once again, we're bouncing around here with energy. It goes to P6A, and then P680 then transfers energy to the reaction center. And it's at this point that we get our first electrons. Now, those electrons, while we're giving the electron here, it actually doesn't, that original electron does not directly come from the reaction center. It is going to come from water. But we'll come back to that in just a moment. Okay, I think I misspoke here. Eventually it comes from water. Our light harvesting complex is now missing an electron. So we need to get that electron back. That comes from water. And so that comes from a protein known as the oxygen evolving protein. This comes from nuclear DNA. So this contains manganese, chloride, calcium, and it splits water into oxygen plus protons. And it's going to transfer those electrons back to the P680 to replenish the electrons that we lost there. So the ultimate source of the electrons that we get is coming from water. And it keeps replenishing the electrons that are coming from the P6A and reaction center complex. And then those get donated again, and it replaces from water. Does that make sense? Okay. So back here with this, here's what we get, is we get light interacting with photosystem 2, which is a light harvesting complex. We're going to have energy passing around. And here is going to then that would be up here, interact with the reaction center in P680. They're going to raise electrons into a higher orbital here, and then those get transferred to Plastoquinone. At the same time, we have the oxygen evolving complex down here where we take water, and it's going to split that into oxygen plus a proton, or a couple protons. And those spare electrons then get redonated to the reaction center up here, and we now have this kind of fully completed. A nice side to this is down here, the proton that we have generated is now ready to increase the amount of protons that we have inside the thylakoid lumen. So when we're talking about electron transport chain, the only way in which we're getting protons inside the matrix is to pump them. In photosynthesis, we're actually getting them two ways. We will talk about pumping protons across this, but we're also generating them from water as well. So plastoquinone is similar to ubiquinone that we talked about in electron transport chain, in that this is a lipid molecule. One difference is that this can transfer 2 electrons at a time, not just a single electron. Yes?
You mentioned earlier how the proteins are super mobile, so what's the need for an intermediary in the step if they could just like bump into each other and directly transfer the electrons?
My guess is that even though they're very mobile, they're still not as mobile as this is, and so this still increases efficiency. But that is an educated guess. And it's transferring between photosystem 2 down here, and I'll transfer it to a cytochrome that lies beyond that. This is on the stromal side of the membrane. Tends to be on the stromal side. And again, like ubiquinone, it is hydrophobic and very mobile in the membrane. The membrane is very fluid anyways, but this gets through it even faster than it would otherwise. So this passes a couple of electrons on to cytochrome B6F, or actually a electron onto cytochrome B6F complex. And this is similar to the mitochondrial cytochrome BC1. So again, we're sitting right here. This has 4 polypeptide chains, 1 from nuclear DNA and 3 from the chloroplast DNA. And it contains iron in a heme group. This is again similar to what we were saying with the cytochromes that we talked about in electron transport chain to mitochondria. And an iron sulfur polypeptide. This is going to transfer an electron from plastoquinone to plastocyanin that we're going to talk about in just a moment. And for each proton, or each electron that we transfer, we're going to pump 2 protons from the stroma into the lumen of the thylakoid. Then it goes on to plastocyanin, where we're only transferring 1 electron at a time. And plastocyanin is here. It's going to pass between cytochrome B6F onto photosystem 1. Plastocyanin is named cyan because it contains a copper molecule. And it goes between 2 different oxidation states, from copper 3+ to copper 2+. Now, plastocyanin is interesting. I talked about earlier that not all of the electron transport chain items in the electron transport chain for photosynthesis are actually bound in the membrane. And this is what I was talking about. Plastocyanin is free in the thylakoid lumen. And it transfers an electron from cytochrome B6F to photosystem 1 reaction center. Now, like Plastoquinone, this is very mobile, and so it can transfer electrons and electron very efficiently. Which leads us into Photosystem 1, which I will talk about more on Monday when we come back. But with that, enjoy. It's Wednesday, right? You gotta say enjoy your weekend already. Enjoy your weekend. Don't come because I'm going to be at Murdoch, and instead you're going to have a video that you watch. So that'll be posted up on D2L.